There are times when we need to store plant parts other than seeds to ensure that we preserve diverse varieties of crops. Seeds don't actually capture the specific varieties of many of our fruit and nut crops. For example, if you plant the seeds of a Honeycrisp apple, the resulting tree will not produce fruit that are the Honeycrisp variety. This is because the fruit itself grows from maternal tissues on the Honeycrisp tree, and the seeds are a result of fertilization events using pollen from another apple variety. Apples and most tree fruits and vines, as well as nuts and tropical fruit crops, are propagated by methods such as cuttings or grafting rather than seeds. Because of this, we need to conserve non-seed tissues of the specific varieties. Our National Gene Bank system has about 30,000 varieties of 89 fruit and nut crops, as well as some vegetables, such as garlic and sweet potatoes, that may need to be stored as something other than seeds at the National Laboratory for Genetic Resources Preservation. Most of these plants are grown in the field locations all over the United States, and these field collections are susceptible to diseases, extreme weather, and natural disasters. Therefore, it's particularly important that these collections be secured here at NLGRP. With our collaborators, NLGRP research programs have developed methods to cryopreserve buds from dormant twigs, as well as 1 mm shoot tips from garlic, citrus twigs, and many kinds of plants grown in tissue culture. Here's an example of grapes and kiwi fruit that we will be cryopreserving from these tissue culture plants. We need to remove the free water for successful cryopreservation of plant tissues. Otherwise, ice crystals form when we freeze the material, and that kills the plant cells upon rewarming. We'll use the apple collection as an example to explain the cryopreservation of a fruit crop collection in the form of dormant buds. There are 2,569 field trees in the USDA-ARS National Plant Germplasm System collection in Geneva, New York. 1,273 of these are different varieties of dessert and cider apples. The rest are ornamentals, such as crab apples and wild species. First, twigs are collected from trees in our National Apple Collection in January. We need them to be as dormant as possible so that the dormant buds can survive the drying and cooling steps. After the twigs are harvested, they are shipped to Fort Collins. They are then cut into short segments, each containing a dormant bud, which will grow into a shoot after cryopreservation. The segments are then dried to about 30% moisture content, packaged into tubes with 10 bud segments per tube and slow cooled to -30 degrees Celsius, at which time they are plunged into liquid nitrogen. The tubes are then transferred to the cryotanks in our vaults for long-term storage. We have about 2,000 different kinds of apple trees backed up in liquid nitrogen at NLGRP, and we cryopreserve them as dormant winter buds. The cryopreservation methods were developed by USDA scientists and their colleagues at Colorado State University over 30 years ago. We have 5 cryotanks that store the dormant buds in the vapor phase of liquid nitrogen, which is about -185 degrees Celsius. It's particularly important that we back up the apple collection at NLGRP because the field collection is vulnerable to threats that include fire blight, as well as waterlogged soils. We can use the same method for other temperate woody crop collections that are in cold hardy locations in our National Gene Bank system. When the curation team in Geneva needs a new variety to replenish a tree in the field, they can request a tube of dormant buds of that variety from NLGRP. A tube of dormant buds is removed from liquid nitrogen, warmed, and rehydrated in moist peat moss. After a week or two, the buds can be grafted onto apple rootstocks and allowed to recover in the greenhouse. When the trees are established, they are planted in the field collection. The USDA-ARS National Apple Collection is an important global resource for apple genetic diversity. Researchers and plant breeders use materials in the collection for their programs. The heritage trees in the collection have also been used to help identify unknown apple varieties throughout the United States, providing a more complete picture of the diversity we need to capture. It's important that we provide a security backup of our apple collection, as well as other vegetatively propagated collections in our national gene bank. We do this to preserve varieties of crops that are not usually propagated by seed, as in the case of apples.